So I guess we can present what we've been working on. Informally, we've called the project NexusSeq, and what we're trying to do is bridge our preclinical experiments to the existing body of literature. So the first problem we really had with this was that when we look at gene expression in model organisms, we can't necessarily make a one-to-one mapping to humans because a lot of times there are genome duplications, and you may have like a many-to-one or one-to-many ortholog mapping situation. So the solution that Gerkelmo and I came up with was abstracting to the level of gene sets or pathways. So instead of saying that, the ABCA1 transporter is upregulated, we can say that broadly ABC transporters are upregulated. And so we took all of our data sets, mouse, zebrafish, and fly, and we got rankings of pathways and gene sets differentially expressed. And then we ran the similar analyses with public data. So a lot of it's from Target ALS and ALS Consortium. We also have some frontotemporal dementia data. And we thought that by comparing on the landscape of the pathways, we'd be able to see how our drugs stack up against the existing market and what we know about these two diseases. So I can share my screen and show you what we have so far. So like I said, we've called it NexusSeq. So a couple weeks ago, maybe 2 weeks ago, we finished this database here. And this pulls those pathways in from a bunch of different studies. So for example, this one here is one from us, Canurta. It's in Daniel, which is the zebrafish. So I'm trying to move this window out of the way. And it shows for this pathway all the genes involved in it. And we can also pull how significant this pathway is. So this score here is how significant that pathway is. This is pretty largely upregulated. And then within this database, Gerkelmo, do you know how many rows we have?
How many what?
Rows, like how many entries we have in this?
I think like 87,000.
So there's 87,000, which comes from a lot of different data sets. So I think we can filter here. For example, we can take a study from these guys. This guy looked at just ALS, mainly from Target ALS, and you can see those same pathways here and how upregulated or downregulated they were in that study. And so this was mainly like the back end, and this allows us to do searching, filtering for all these different pathways. And this worked out so well that we wanted to then expand on it because of how successful it was. And so now instead of just taking results that other people have published, we wanted to work with their raw data from supplementary files and run our own internal analysis on their data to then make sure it's the same, up to the same standards. So that's something that Gerkelmo's mainly been putting together on AWS. Do you want to present that? Okay, all right. Just regarding that last piece, the pipeline, this is kind of what I've really been focusing on the past year I've been working here, right, is analyzing this RNA sequencing data. If you guys remember, like at the beginning when I first started, like last February, I did the analysis of the zebrafish data set, and that took about 2 months. And then the Drosophila data set, that took Gerkelmo and I together about a month. I was then able to do the mouse study in like a week. And the reason that it got so much faster was because of the amount of things that I learned and how efficient I got with the process. And I think that's something that's actually lacking a lot in industry. Like I was looking at some papers where the majority of people just kind of skip by steps and don't really give attention to a lot of these things. And it's just because it's such a difficult task to do. And then you see a lot of errors, like we saw with gene markers, where they don't really know what they're doing, to be honest. And so that's why I wrote that report that culminates everything that I learned, and that leaves it all there on the table. And that's what Gerkelmo and I are building from, so that we can have a not fully automated, but largely automated pipeline with availability for us to intervene where we need. And the goal is to be able to take all this public data, process it, and add it to this growing database. And the reason why we want to add it to this database is so we can do cool things like this. This is something I just made this week very quickly. This will come up here in a second. So this is comparing CFA and CNR 401 to the literature. So first I did an ALS analysis. So this ALS background represents, I don't know how many pathways. I think Gerkemel said it's like however many thousand, but it's all these pathways that are implicated in ALS versus healthy control. Here, actually, it tells you 5,135. So these are all pathways that are implicated in ALS. And what I've done here is show a bar graph showing what our drug can reverse. So this is how many of those pathways are reversed in CFA treatment. And you can see if this is the total 100%, how many CFA is able to reverse. Now, this doesn't show magnitude or statistical significance. It's very rudimentary, but it gives you an idea of what the effective CFA is. And then we have CNR 401. And I was also able to find RNA sequencing from Adaravone, and I included that as well. So this replicates what we actually saw in the zebrafish paper from a year ago, where CFA has a really large transcriptomic impact. That doesn't necessarily correlate to better therapeutic potential, but it is interesting to see how many of the disease pathways we're able to reverse. And then here I just visualize that a little bit differently. So where 100% of this pie chart is all these disease genes, this is how many CFA is able to reverse, and then the same for CNR 401. So that's an example of what we can pull once we have a lot of data in this database. This is another really cool thing that we were able to do. So this is specifically C9 ORF72 ALS. So that's a specific genetic genotype of ALS. And the background for this is a data set where the person actually did single cell sequencing, so at the resolution of single cells, and then grouped those single cells into groups such as neurons, oligodendrocytes, and other glia in the frontal area of the brain and also the motor cortex of the brain. And so I remembered that our Drosophila data was also a C9 data set. So this is specifically comparing Drosophila to that data set. And this is the amount of pathways we were able to reverse the regulation of. And this just kind of helps us see where our drug is actually having an impact. Because we did do bulk RNA-seq in the brains of Drosophila, but this can kind of give us an indication that we're mostly affecting other glia, which makes sense, right? Because we're not directly affecting the motor neurons through proteomics, we're more affecting the endogenous support pathways of the organism. And then I did the same thing here for dementia. And of course, as this database scales, we could add in whatever disease we wanted. So that's what we've been working on the past couple of weeks.